Hello, can I
Please
start?
start, madam.
Okay. Good evening. We will continue the next part of inflammation. Today we will learn about phagocytosis.
Please start it, madam. Please start it.
Yeah. So let's go through a quick recap of what we did in the last class of inflammation, because phagocytosis is the next step of whatever we learned in the last class. So what we learned was the vascular events in acute inflammation, saying that vasodilatation induced by chemical mediators such as histamine and serotonin is the cause of erythema and stasis of blood flow. Increased vascular permeability is induced by histamine, kinins, and other mediators, which will produce gap between the endothelial cells by direct or leukocyte-induced endothelial injury and by increased passage of fluids through the endothelium. Then there was increased vascular permeability, which allowed the plasma proteins and the leukocytes, all of this to leak out into the site of inflammation, and that the leukocytes move towards tissue damage. And this fluid leak from the blood vessels resulted in edema. Then there was lymphocytic vessels and lymph nodes, which are also involved in inflammation, and they often show redness and swelling. We also learned about the leukocyte recruitment to the site of inflammation, how it happens. We learned that from the blood vessel, the leukocytes will move towards the site of injury by a multi-step process which has rolling, attachment. There are a lot of cytokines which promote the selectins and integrins, which are the ligands on the endothelium, and how through these integrins the leukocytes will move out of the blood vessel and then are pulled towards the chemotactic stimulus, that is the site where the tissue injury has occurred. And we also learned that neutrophils are the main leukocytes in the early inflammatory process, and later they are replaced by monocytes and macrophages. So this was a quick recap of the acute inflammation class which we did last time. So today's topic is phagocytosis. Now, after the leukocytes have managed to get out of the blood vessel and come into the area where there is tissue injury, basically what is going to happen is that the offending agent is going to be killed, and that is achieved by the process of phagocytosis. So the process of phagocytosis has three sequential steps. One is the recognition and attachment of the particle to be ingested by the leukocyte. Second, after it gets attached, it is going to be engulfed. That's called engulfment. How with the subsequent formation of a phagocytic vacuole. And after that, whatever has been ingested is going to be killed and degraded. So these are the three steps of phagocytosis. Now, first step, whatever needs to be killed will have to come and attach to the receptor. That is called as the phagocytic receptors. There are different kinds of phagocytic receptors. Some of them are mannose receptors, scavenger receptors, and receptors for various opsonins. These will bind and they will ingest the microbes. Let's see each one of them. Mannose receptor, basically it's the macrophage mannose receptor, which is a lectin. Basically, it binds the terminal mannose and fucose residues of the glycoproteins and glycolipids. And these sugars are typically part of the molecules which are found on the microbial cell wall, whereas mammalian glycoproteins and glycolipids basically contain terminal sialic acid or N-acetyl galactosamine. So this is basically the chemical composition of biochemical composition of the cell wall. So this is how the leukocyte is able to differentiate that it is going to attack and kill only the microbes and not the host cell. So what is there? The microbial cell wall, they are rich in the mannose, terminal mannose and fucose residues. Whereas the mammalian glycoproteins, they are the terminal sialic acid and N-acetyl galactosamine. That's how they will differentiate. Second kind of receptors are called as the scavenger receptors. What are the scavenger receptors? They were originally defined as molecules that bind and mediate the endocytosis of oxidized or acetylated low-density lipoprotein particles that can no longer interact with the conventional LDL receptor. So basically, they were there to scavenge and remove, which is no longer needed for the body. There are also certain macrophage scavenger receptors which will bind a variety of microbes in addition to these modified LDL particles. Thus, they will participate in phagocytosis. Apart from this, we have macrophage integrins, mainly the MAC1, which will also bind microbes for phagocytosis. The efficacy of phagocytosis is greatly enhanced when the microbes are opsonized by specific proteins. They are called opsonins. Basically, it's going to be made tasty so that the leukocytes are going to engulf them easier. Okay, so that is basically what we call as high affinity receptors. Then the major opsonins are IgG antibodies, C3b breakdown product of complement, and certain plasma lectins, mainly the mannose binding lectin, all of which are recognized by specific receptors present on the leukocytes. Whenever I talk leukocytes, what do I mean in the process of phagocytosis? Initially, it is neutrophil. But in many tissues, it is monocyte, which will become macrophage. So these are the three main cells involved in phagocytosis. So this is a pictorial representation of the kinds of different classes of cell surface receptors. You know, last time we learned about this, isn't it? So the G protein coupled receptors, which is going to be present on the leukocyte membrane. Once the microbes are recognized and bound to this, it will cause cytoskeletal changes and signal transduction, which will increase the integrin avidity. Remember the vascular events, and thus it will increase the adhesion of the leukocytes to the endothelium. Apart from this, it also causes chemotaxis. Thus, it will enhance the movement of the leukocyte towards the tissues, that is migration into the tissues. That was one class of receptor. Second class of receptor is the toll-like receptor, which will bind the LPS polysaccharide residue on the microbe, and after which it will result in the production of various chemical mediators of inflammation, such as arachidonic acid metabolites and cytokines. We will learn about all of these, which will result in amplification of inflammatory reaction, and so that the offending agent is removed as early as possible. Along with this, today we are going to learn about these phagocytic receptor, which I just listed out, which were the different categories of the phagocytic receptors. So the microbe is going to bind to the phagocytic receptors, and once that binds to it, there is going to be two things which will happen. One is production of reactive oxygen species and lysosomal enzymes which are released. Along with that, this will facilitate the phagocytosis of the microbe into the phagosome. That will result in the microbicidal activity of the leukocytes with the ultimate killing of the microbe. So that's how the whole process starts, and this is how the different receptors on the membrane of the leukocyte participate in from the recognition till the final killing of the offending agent. Commonly, example is a microbe. So after the microbe, let's take the example as microbe. After the microbe is now sitting on the membrane of the leukocyte, it is now recognized, it is sitting there. Now what will happen? Next thing is that it has to be taken within the cell. That process is called as engulfment. After a particle is bound to the phagocyte receptors, extensions of the cytoplasm, which are also called the pseudopods, they will flow around that particle, and the plasma membrane pinches off to form a vesicle. Now that vesicle, which encloses that microbial particle, is called as a phagosome. Then you know that it is a cell, it's a leukocyte, it has a lysosome within it. We are talking of neutrophils and macrophages. So they have lysosomes in it. So this phagosome will fuse with the lysosome. which will result in the discharge of this lysosomal granules into the phagolysosome. Now, during this process, the phagocyte may also release granule contents into the extracellular space. The process of phagocytosis is complex, involving the integration of many receptor-initiated signals, which will lead to membrane remodeling and cytoskeletal changes. And phagocytosis is dependent on the polymerization of actin filaments. So that's the cytoskeletal change which will happen within the leukocyte cell membrane for the process of phagocytosis to take place. Now, once the phagosome is formed, the phagolysosome is formed, and there is release of these granules, and with that release of granules, there's going to be reactive oxygen species. Finally, the microbe gets killed. Now, the intracellular destruction of microbes and debris basically is accomplished by the reactive oxygen species and reactive nitrogen species, mainly derived from nitric oxide. And these, as well as the lysosomal enzymes, will destroy the phagocytosed debris. So, let's learn a little in detail about the reactive oxygen species. See, the reactive oxygen species are basically produced by the rapid assembly and activation of a multi-component oxidase, which is the NADPH oxidase, which will oxidize the NADPH, and in the process, it will reduce the oxygen to superoxide anion. Now, in the neutrophil, this oxidative reaction is triggered by activating signals and accompanies phagocytosis, and it is called as the respiratory burst. Now, this is very important step in the killing of the microbe, respiratory burst, which is basically the release of the superoxide anion. Phagocyte oxidase is an enzyme complex consisting of at least 7 proteins, if we look at it the structural way. Now, normally what happens is, in the resting neutrophils, different components of the enzyme are located in the plasma membrane and the cytoplasm. In response to activating stimuli, the cytosolic protein components will translocate to the phagosomal membrane, where they assemble and they will form the functional enzyme complex. So, this will result in the reactive oxygen species, which are produced within the lysosome and the phagolysosome, where they will act on the ingested particles without damaging the host cell. Even though the enzyme is present, it's not in its activated form. So, oxygen is then converted to hydrogen peroxide, mostly by spontaneous dismutation. And hydrogen peroxide, that is H2O2, is not able to efficiently kill the microbe by itself. So what will happen is, the azerophilic granules which are present within the neutrophils, they contain an enzyme called as myeloperoxidase. That's why the neutrophilic series are called granulocytic series. If you remember the nomenclature, they contain granules. And what is the function of the granules? That is what we are learning today, that it is basically to kill the microbes. So this myeloperoxidase enzyme, which in presence of the halide ion such as chloride, will convert the hydrogen peroxide to hypochlorite. Now this hypochlorite is a potent antimicrobial agent which will destroy the microbes by halogenation or by oxidation of proteins and lipids. So that is how reactive oxygen species will kill the offending agent. So this H2O2 MPO halide system, that is hydrogen peroxide myeloperoxidase halide system, is the most efficient bactericidal system of neutrophils. H2O2 is also converted to hydroxyl radical, another powerful destructive agent. So these oxygen-derived free radicals will bind to and modify the cellular proteins, cellular lipids, and the nucleic acids, and thus will destroy the cells such as the microbes. Oxygen-derived radicals may be released extracellularly from the leukocytes after exposure to microbes, chemokines, and antigen-antibody complexes, or following a phagocytic challenge. These reactive oxygen species are implicated in the tissue damage accompanying the inflammation. So what happens is, apart from being released into the phagolysosome, some amount of these reactive oxygen species radicals, free radicals, do also get released into the extracellular environment like a collateral damage. And that will result in some amount of normal tissue being damaged in inflammation when the body is trying to get rid of the offending agent. The other important chemical which is involved in phagocytosis is nitric oxide. It's a soluble gas produced from arginine by the action of nitric oxide synthase, which also participates in microbial killing. Now, if we think about it, there are three different types of nitric oxide synthase. One is an endothelial nitric oxide synthase, the other one is neuronal, and the third one is inducible nitric oxide synthase. Now, the endothelial and the neuronal ones are expressed at low levels, and the nitric oxide they generate functions mainly to maintain the vascular tone in case of endothelial NOS, and as a neurotransmitter in case of neuronal NOS. Whereas the inducible NOS is the one which is involved in microbial killing, and it is induced when the macrophages are activated by cytokines such as interferon gamma or microbial products. And in macrophages, the nitric oxide reacts with superoxide to generate the highly reactive free radical peroxynitrate. These nitrogen-derived free radicals, similar to the reactive oxygen species, attack and damage the lipids, proteins, and nucleic acids of the microbes and the host cell. Thus, it will kill the microorganism, offending organism, causing some amount of damage to the host cell as well. So, if we have to describe pictorially what we just learned, first step was the recognition and attachment of these microbes to the phagocyte receptor. Once it is attached, there are these cytoskeletal changes which happen, which will result in the formation of pseudopods and pinching off of the cell membrane around the offending microbe, forming a phagosome. So this becomes a phagosome, which is represented here. And we already know that there is a lysosome present within the cell. Now, this lysosome is present in the inactivated form. Once the microbe gets attached here, this will move on to the inner part of the cell membrane and will get activated with the active enzymes present in the lysosomes. Now, this lysosome and phagosome will fuse, will result in the formation of phagolysosome. Now, what happens once a phagolysosome is formed? We can see that the granules which were present in the lysosome are released, and these granules release something called as an oxidative burst. What is an oxidative burst? Which is typically seen in the neutrophils. Now that results in the cleaving of oxygen into reactive oxygen species. Okay, then we have an enzyme called as MPO within the neutrophils. So the hydrogen peroxide which is present within the granules of the neutrophils are going to be cleaved into the superoxide radical, which will result in the destruction of the microbe. So along with the reactive oxygen species or the oxidative burst, we also have nitric oxide acting. The inducible nitric oxide synthase is going to produce the nitric oxide from arginine, which will result in, this will get cleaved, and it will result in the formation of the reactive peroxynitrite, which is going to altogether are going to degrade the microbe and they're going to damage the cell wall. Because of this, because they're going to act on the lipids, the proteins, and the nucleic acid, and the offending agent is killed. So this is the whole process of phagocytosis. Lysosomal enzymes and other lysosomal proteins. So on a broader sense, we learned how phagocytosis happens with the reactive oxygen species, the nitric oxide. Now, apart from these two, there are also certain other lysosomal enzymes and proteins which will contribute to the killing of the offending agent. Let's list them. Neutrophils and monocytes contain the lysosomal granules that contribute to the microbial killing, and when released will contribute to the tissue damage. Neutrophils have two main types of granules. One is the smaller specific granule contain lysosome, collagenase, gelatinase, lactoferrin, plasminogen activator, histaminase, and the alkaline phosphatase. Okay. Then you have the larger granules, also called azerophil primary granules. They contain the myeloperoxidase, bactericidal factors, the lysozyme, and the defensins, acid hydrolases, and a variety of neutral proteases such as the elastase, catepsin G, non-specific collagenases, and the proteinase 3. Both types of granules can fuse with phagocytic vacuoles containing engulfed material, or the granule contents can be released into the extracellular space. Now, different granule enzymes serve different functions. For example, the acid proteases will degrade the bacteria and the debris within the phagolysosome, which are acidified by the membrane-bound proton pumps. Then you have the neutral proteases, which are capable of degrading various extracellular components, such as the collagen, basement membrane, fibrin, elastin, and cartilage, which will result in tissue destruction, which also accompanies the inflammatory process. The neutral proteases can also cleave the C3 and C5 complement proteins directly, which will yield anaphylatoxins and will release a kinin-like peptide from the kininogen. Then we also learned about elastase. So what will elastase do? It's been shown to degrade the virulence factor of the bacteria and thus will combat the bacterial infection. Macrophages also contain acid hydrolases, collagenases, elastases, phospholipases, and plasminogen activator. So these are the different lysosomal proteins present within those granules. Because of the destructive effects of the lysosomal enzymes, the initial leukocytic infiltration, if unchecked, can potentiate further inflammation by damaging the tissues. So it's a double-edged sword. If the balance is tipped in the other way, it is harmful to the body as well. These harmful proteases, however, are normally controlled by a system of anti-proteases in the serum and in the tissue fluids. Foremost among these is the alpha-1 antitrypsin, which is the major inhibitor of neutrophil elastase. And you may know that a deficiency of these inhibitors will lead to sustained action of leukocyte protease, which is a case in patients with alpha-1 antitrypsin deficiency. This is the basic pathogenesis in patients with alpha-1 antitrypsin deficiency, which will lead to the whole lot of serious clinical outcome. Apart from the lysosomal proteins and the lysosomal enzymes that we learned, there are other microbicidal granule contents. These include the defensins, the cationic arginine-rich granule peptides that are toxic to the microbes. Then you have cathelicidins, antimicrobial proteins which are found in the neutrophils and other cells. The lysozyme, which hydrolyzes the muramic acid N-acetyl glucosamine bond found in the glycopeptide coat of all bacteria. Then you have lactoferrin, which is an iron-binding protein in specific granules. Then there is major basic protein, which is a cationic protein of eosinophils, which basically has limited bactericidal activity, but it is cytotoxic to many parasites. So you can see that in parasitic infestation, the marker of parasitic infestation on the peripheral blood is eosinophilia. So high levels of eosinophils are produced to get rid of the parasites. Then there is one more terminology called as neutrophil extracellular traps, NET, not the NEET for which you are studying. This is NET. What does it mean? Neutrophil extracellular traps are basically the extracellular fibrillar networks which provide a high concentration of antimicrobial substances at the site of infection, and they prevent the spread of microbes by trapping them in these fibrils. They are produced by the neutrophils and in response to the infectious pathogens, mainly the bacteria and the fungi, and the inflammatory mediators such as chemokines, cytokines, complement proteins, and the reactive oxygen species. The extracellular traps, they're basically not within the cell, they're extracellular. It's like this whole meshwork, like I can give you an example of how a spider will build a mesh kind of a trap, to trap the insect. So this is that kind of a trap which consists of a vicious meshwork of nuclear chromatin, and it will make sure that whatever left out microbes are there are going to be trapped within this meshwork, and concentrates granule proteins such as antimicrobial peptides and enzymes. So to show you an example, this is how neutrophil extracellular trap looks like. This is a fluorescent microscopic picture of a neutrophil. See, this is a normal neutrophil which has a red colored nucleus. Sorry, the nucleus is red and the cytoplasm is green, if you can appreciate here. Yeah, there are 3 of them. Then what happens, this is the release of the nuclear material from the neutrophil. They have lost all the nucleus, so all this red nuclear material is all released into the extracellular space, and this is what is forming an extracellular trap. Now if you see the electron microscopic picture of this is how it looks. This is all their trap. This is the extracellular trap of the nuclear material of the neutrophils. These are the bacteria. So this is basically the bacteria. Usually the one which is represented here is a staphylococcus. It's a cocci, round shaped bacteria which is trapped in this meshwork of neutrophil extracellular trap. Yeah. In the process of NET formation, the nuclei of the neutrophils are lost, leading to the death of the cells. And the NETs have been detected in the blood during sepsis, and it is believed that their formation in circulation is dependent on platelet activation. The nuclear chromatin in the NETs, which will include the histones and associated DNA, have been postulated to be a source of nuclear antigen in systemic lupus, systemic autoimmune diseases, particularly the lupus, in which individuals basically react against their own DNA and nucleoprotein. So this was something which was devised to get rid of the offending organism, but sometimes because there is also collateral damage, there are certain autoimmune diseases which will develop, and it is postulated that this is one of the mechanisms where, particularly in lupus, this is what is the pathogenesis behind it. This is about neutrophil extracellular trap. So we now discussed as to how phagocytosis is very important to get rid of the offending agent, and how our leukocytes are the ones which will phagocytose the offending agent. Apart from this, there is always a collateral damage. So there is some amount of leukocyte mediated tissue injury. Okay, so that causes injury to normal cells and tissues under several circumstances. Some examples are as a part of normal defense reaction against infectious microbes when adjacent tissues suffer collateral damage. In some infections that are difficult to eradicate, such as tuberculosis and certain viral diseases, the prolonged host response contributes more to the pathology than does the microbe itself. So, which is the commonest right now, we are seeing something which is causing this, something causing cytokine storm being talked about in everyday newspaper is the COVID. The COVID also, the pathogenesis of COVID also is something like this. So whatever the body produces or mounts an immune response against the offending agent, later it becomes and it goes so unchecked that there is a cytokine storm which is going to eat away the normal host tissues. So this is one mechanism by which COVID pathogenesis is being explained. The other example where leukocyte-mediated tissue injury happens is when the inflammatory response is inappropriately directed against the host tissues like in autoimmune diseases, like we discussed for lupus. When the host reacts excessively against usually harmless environmental substance, as in allergic diseases, including the asthma. So what is the mechanism of this tissue injury? In all these situations, the mechanism by which the leukocytes damage normal tissues are the same as the mechanisms involved in antimicrobial defense, because once the leukocytes are activated, their effector mechanisms do not distinguish between the offender and the host. During activation and phagocytosis, neutrophils and macrophages produce microbicidal substances, that is, the reactive oxygen species, the nitric oxide, and the lysosomal enzymes within the phagolysosome. These substances are also released into the extracellular space. These released substances are capable of damaging the normal cells as well as the vascular endothelium, and thus may amplify the effects of the initial injurious agent. If unchecked or inappropriately directed against the host tissues, the leukocyte infiltrate itself becomes the offender, and indeed the leukocyte-dependent tissue injury underlies many acute and chronic human diseases. So, we just discussed about the mechanism of leukocyte-mediated tissue injury. The contents of the lysosomal granules are secreted by leukocytes into the extracellular milieu by different mechanisms. Controlled secretion of the granule contents is a normal response to activated leukocytes. But what happens is, if the phagocytes encounter materials that cannot be easily ingested, such as these immune complexes deposited on an immovable flat surface, especially the glomerular basement membrane, the inability of the leukocytes to surround and ingest these substances, that's called as frustrated phagocytosis. Now, this particular thing will trigger strong activation and release of large amounts of lysosomal enzymes into the extracellular environment. Some phagocytosed substances such as urate crystals may damage the member of phagolysosome and also lead to the release of lysosomal granule contents. So this is where one or two of the examples where the leukocyte-mediated tissue injury itself is going to become the disease. Apart from this, there are certain other functional responses of the activated leukocytes, apart from killing the offending organism. Now, in addition to eliminating the microbes and the dead cells, the activated leukocytes play several other roles in the host defense. Importantly, these cells, especially the macrophages, will produce the cytokines, which can either amplify or limit the inflammatory reactions, growth factors, which stimulate the proliferation of endothelial cells and fibroblasts, and the synthesis of collagen and enzymes that remodel the connective tissue. So basically, depending upon the offending agent, it will either amplify or it will reduce the inflammatory reaction and secretion of all growth factors and synthesis of collagen and enzymes. Because of these activities, macrophages are also critical cells of chronic inflammation and tissue repair after the inflammation has subsided. So once inflammation goes, next the macrophages will go into something called as healing. That we will discuss subsequently. Now, all these while we spoke about mainly neutrophils, monocyte and macrophages as being the cells in acute inflammation to get rid of the offending agent or phagocytosis. Now, recent theories have postulated that T lymphocytes, which are the cells of adaptive immunity, also contribute to acute inflammation. The most important of these cells are those which will produce cytokine interleukin 17, which is an important mediator of inflammation, acute inflammation. Also, T lymphocytes will produce interferon gamma, which will activate the monocyte macrophage system. So basically, even T lymphocytes are now believed to be involved in the process of acute inflammation, though not directly, but by the production of interferon gamma and cytokine interleukin 17. So after the phagocytosis is done, how will the acute inflammatory response be terminated? Now, such a powerful system of host defense with its inherent capacity to cause tissue injury needs very tight control to minimize the damage. In part, the inflammation declines after the offending agents are removed simply because the mediators of inflammation are produced in rapid bursts only as long as the stimulus persists. They have short half-lives and they are degraded after the release. Neutrophils also have short half-lives in tissues and they die by apoptosis within a few hours after leaving the blood. So this is all when normal things are happening, okay? In addition, as the inflammation develops, the process itself will trigger a variety of other stop signals that actively terminate the reaction. And these active termination mechanisms include a switch in the type of arachidonic acid metabolite produced from pro-inflammatory leukotriene to anti-inflammatory lipoxin and the liberation of anti-inflammatory cytokines, including the transforming growth factor beta and interleukin 10 from macrophages and other cells. Other control mechanisms that have been demonstrated experimentally include the neural impulses, which will inhibit the production of TNF in the macrophages. So these are the various mechanisms. The acute inflammatory response is terminated. So from today's class, what is the take-home message? Mainly about the leukocyte activation and removal of offending agent by phagocytosis. So what did we learn? This is the gist of whatever I have spoken till now. The gist is that the leukocytes can eliminate microbes and dead cells by phagocytosis, followed by their destruction in the phagolysosome. Destruction is caused by free radicals, that is reactive oxygen species and nitric oxide, which is generated in activated leukocytes and lysosomal enzymes. Neutrophils can extrude in their intracellular contents into the extracellular nets that will trap and destroy the microbes. Then we also learned that enzymes and reactive oxygen species may be released into the extracellular environment. And the mechanisms that function to eliminate the microbes and the dead cells are also capable of damaging the normal tissues in the pathologic consequence of inflammation. And the anti-inflammatory mediators terminate the acute inflammatory reaction when it is no longer needed. So we learned about all this in detail. Let's move on to the MCQs. In an experiment, I just want to know, do we have students on the call, on the live session? Do we have students on the live session, please? No? I just wanted some interaction. Do we have students on live session? Okay, I will just continue then. I thought an interaction would be better. If there are no students, I will just give you the answers. It's difficult for me to clarify your doubts if you have any. If you have doubts, you can please speak up. MCQ is in an experiment, neutrophils collected from the peripheral blood are analyzed for a burst of oxygen consumption. Now, this respiratory burst is an essential step for which of the following events in an acute inflammatory response? The question is straightforward, but just look at the options. Attachment to the endothelial cells, generation of microbicidal activity, increased production in the bone marrow, opsonization of bacteria, phagocytosis of bacteria. So, do we have any givers for the answer? The respiratory burst basically is for the killing of the microbes. So the answer is generation of microbicidal activity. Now there is one more option which is confusing here, which says phagocytosis of bacteria. See, generation of microbicidal activity by the respiratory burst is a part of phagocytosis of bacteria. Since this answer is more accurate, and this is a broader answer, I have chosen generation of microbicidal activity as the answer to this particular MCQ. If you didn't have this particular option, the answer would be phagocytosis. It's not opsonization. It has nothing to do with increased production in bone marrow. And obviously, respiratory burst doesn't have any role in attachment to the endothelial cells. So it is basically under phagocytosis. But since the exact step under phagocytosis is given in the option, we have chosen generation of microbicidal activity. Next question. A 4-year-old girl has had numerous infections with Staphylococcus aureus since infancy. Genetic testing shows a defect leading to lack of beta-2 integrin production. Which of the following abnormalities of neutrophil function is most likely responsible for these clinical symptoms? Right? So they have very clearly given that there is a lack of beta-2 integrin. So where did we learn about integrins? Let's see the options. Is it because of decreased generation of hydroxy halide radicals? Is it because of diminished phagocytosis of bacteria opsonized with IgG? Is it because of failure of migration to the site of infection? Or inadequate adhesion on cytokine activated endothelium? Or is it reduced respiratory burst after phagocytosis? Anybody wants to take? So this is the recap which we did in the first 5 minutes of the session, and the details of this was in last class, right? So it's because of inadequate adhesion on cytokine activated endothelium. So integrins are very important for the leukocytes to bind to the endothelium. From there they will have to sequester out and move towards the site of tissue injury. So selectins were involved in the rolling of the leukocytes, and integrins were involved in attaching of the leukocytes to the endothelium. So a defect in beta-2 integrin production will obviously result in inadequate adhesion of the leukocyte on the cytokine activated endothelium, and that's why the inflammatory process is not going on well, and that's why this girl is having recurrent infections with Staphylococcus aureus. Next question. In an experiment, peripheral blood cells are isolated and placed into a culture medium that preserves their metabolic activity. Interferon gamma is added to this culture along with viable E. coli organisms. Which of the following blood cell types in this medium is the most likely to have bactericidal activity against E. coli? Yeah. We're talking about interferon gamma. Interferon gamma is now acting as a stimulating agent along with the E. coli. So which cell will be attracted towards this chemotactic site? We can imagine it that way. Is it basophil? Is it lymphocyte? Is it CD4 plus lymphocyte? Is it CD8 plus lymphocyte? Is it monocyte? Or is it natural killer cell? Or is it neutrophil? Anybody? So we are giving a chemotactic stimulus with interferon gamma and the microbe. Essentially, there is a microbe and you have added interferon gamma. So interferon gamma activates which cell line? I don't think I have any takers. The answer is monocyte. Basically, the T lymphocyte will produce more of interferon gamma, which is going to activate the monocyte macrophage lineage. So that's what we studied just now. And therefore, in this particular scenario, it will be monocyte because specifically they have given interferon gamma being added to the culture of E. coli. Now here you'll have confusion only between neutrophil and monocyte. Yeah, commonly we could have said it is neutrophil itself, but since they have given us an option that there is interferon gamma in the culture media, we know that interferon gamma activates the monocyte macrophage system. The answer here is the monocyte. Next question. In an experiment, T lymphocytes from the peripheral blood are placed in a medium that preserves their function. The lymphocytes are activated by contact with antigen and incubated for 4 hours. The supernatant fluid is collected and is found to contain a substance that is major stimulator of monocyte and macrophages. Which of the following substances released into this fluid medium is most likely to stimulate the macrophages? See, this question and the previous question are like corollary to each other. Is it histamine? Is it interferon gamma? Is it leukotriene B4? Is it nitric oxide or is it phospholipase C? So basically they have told that there's a T lymphocyte and they are telling that whatever was released from this T lymphocyte, activated T lymphocyte, has stimulated my monocyte and macrophages. So what was released is what they are asking. So previous question, what was the hint? Hint was that interferon gamma was there and therefore which cell is getting activated? So there we said monocyte because interferon gamma was there. Here there is monocyte and macrophage which are already getting activated by something which is released from the T lymphocyte, activated T lymphocyte. What is that something? It is interferon gamma. Yeah, I hope I have made myself clear. T lymphocytes secrete the role of lymphocytes in acute inflammation. When I discussed, I told you that T lymphocytes will secrete interferon gamma and they will also secrete interleukin 12. Right? So interferon gamma is the one which is going to activate the monocyte macrophage system. So that's the whole chronology, how it happens. And these two questions are like complementary to each other. If you know this process and you have the clarity of this concept, you'll answer many more questions related to this. One more. A 12-month-old boy with 6-month history of repeated infections has had fever and cough for the past 3 days. So, you know, he's a very young child, 6-month history of repeated infection. So there's something wrong going on. And what have they done? They saw that a gram stain of the sputum showed many gram-positive cocci in chains. That means it is a staphylococcus. CBC shows neutrophilia. Yes, bacterial infection. Laboratory studies show that the patient's neutrophils phagocytose and kill organisms properly in presence of normal human sera, but not in his own serum. So what does this particular line tell you? Neutrophil number is fine. There is neutrophilia in response to bacterial infection. But when you put the patient's neutrophil in normal human serum, the bacteria are getting killed. But in the patient's serum, when you try to create this atmosphere in the lab, the neutrophils are not killing the bacilli. Now the neutrophils migrate normally in a chemotactic assay. That also they've given you. They've told you there's no problem with the movement of the neutrophil. So basically, where is the mistake? Which of the following is most likely cause of this boy's increased susceptibility to infection? So now we know that there is some problem with phagocytosis in his own serum. So what is lacking in his serum? Is it abnormality of selectin expression? Is it diminished opsonization? Is it defective neutrophil generation of hydrogen peroxide? Is it deficiency of integrins? Is it phagocytic cell intracellular protein defect? So let's rule out one by one. Selectin expression. We are talking here about phagocytosis. So selectin is for rolling. We rule that out. Next is diminished opsonization. What will happen in diminished opsonization? So basically, he is having recurrent infections. So in the normal human serum, the neutrophils are doing fine, whereas his own serum, they're not doing fine. So if the opsonins are not there in his own serum, what will happen? The process will be delayed, because we know opsonization is one important part in phagocytosis. So let's keep that as an option. Let's move ahead to the next option, defective of hydrogen peroxide. So if this was the situation, it would have been that the neutrophils itself are defective, right? So they wouldn't even function in normal serum. So phagocytosis wouldn't have happened. So we'll rule that out. Deficiency integrins. Basically, integrins are for binding of leukocytes to the endothelial membrane. So it doesn't come anywhere in phagocytosis. We rule that out. Last one was phagocytic cell intracellular protein defect. If it was a phagocytic cell intracellular protein defect, then in the serum, in normal human serum, it will not get corrected, because there is a problem within the intracellular protein of the phagocyte itself. Have normalized in the regular serum. So what was the only thing which was provided in the normal serum for the leukocytes to become normal? Opsonins. You know that IgG, complement, all these are opsonins, and they are easily available in the serum. So that's how the deficiency of this patient's opsonins was corrected by the addition of normal human serum, and that's how his phagocytosis improved. Diminished opsonization for this particular question. When we analyze the question, you can come to this conclusion. Last question. A 5-year-old child has recurrent bacterial infections, including pneumonia and otitis media. Analysis of leukocytes collected from his peripheral blood shows a deficiency of myeloperoxidase. A reduction in which of the following process is most likely cause of this child's increased susceptibility to infection? So, recurrent bacterial infection, leukocytes, deficiency of myeloperoxidase, very clearly given. Now, what is reduced? Is it hydrogen peroxide elaboration? Is it hydroxy halide radical formation? Is it failure of migration resulting from complement deficiency? Is it phagocytic cell oxygen consumption? Or is it prostaglandin production? So when we know that myeloperoxidase is difficult, myeloperoxidase is deficient, what is going to happen? That system, the hydrogen peroxide, MPO, halide system, that goes for a toss. So what is the answer? Hydroxy halide radical formation gets affected. And that's why the child becomes more susceptible to infections. Yeah, that's about today's class on phagocytosis. I've tried to elaborate everything and tried to link it with the clinical scenarios so that you will be able to analyze case-based questions and also answer them. Any doubts?
Thank you, madam.
Any doubts? If there are no doubts, we can close the class. Thank you.
Thank you so much, madam. Thank you.
Yeah, thank you.